Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we are beginning Chapter 6, Microbial Culture. A microbial culture is much more than visible growth in a flask or on a plate. It is a controlled biological system in which nutrition, temperature, pH, oxygen, sterility, and time influence which organisms grow and what they produce. We will learn how microbiologists prepare suitable media, prevent contamination, obtain pure cultures, follow a growth curve, calculate viable cell numbers and preserve cultures safely. Throughout the chapter, keep one evidence rule in mind. Growth shows that something grew, but it does not by itself prove culture purity or organism identity. By the end, you should be able to explain each technique, interpret its result, and state its limitation in a CBSE answer. Here is our roadmap. First, we define microbial culture and aseptic technique, then connect microbial metabolism to carbon, energy, nitrogen, minerals, water and growth factors. Next, we classify defined, complex, selective, differential, and enrichment media. We then compare moist heat, dry heat, filtration, radiation and chemical methods, while distinguishing sterilization, disinfection, and antisepsis. After that, we study streaking, spreading, pore plating, and dilution as ways to obtain isolated colonies. We will examine temperature, pH, oxygen and water availability as environmental growth factors, draw the lag, exponential, stationary and decline phases of a batch culture growth curve, and compare direct counts, viable counts, turbidity and biomass. Finally, we calculate colony forming units, discuss culture maintenance, and close with containment, decontamination and biosafety. The central problem is control. Microorganisms are widespread, grow rapidly, and may look similar when observed only as turbidity or colonies. A useful culture therefore needs three kinds of control. Entry control prevents unwanted organisms from entering the vessel and prevents the culture from escaping. Environmental control defines nutrients, temperature, pH, oxygen, and time. Measurement control matches the method to the claim. Optical density estimates light scattering, a plate count estimates viable colony forming units, and dry mass estimates biomass. A negative sterility control can reveal contamination, while repeated isolation and characterization support purity and identity. This framework helps us avoid a common error, treating any visible growth as proof that the intended organism alone is present. Reliable microbial culture joins technique, measurement, comparison, and validation. A microbial culture is a population grown under laboratory conditions in a liquid or solid nutrient medium. Aseptic technique is the set of practices that prevents unwanted microorganisms from entering cultures, people, or the environment. Sterile medium and glassware are prepared before inoculation. During transfer, containers are opened only briefly, sterile loops or pipette tips are used, and the working area is organized to reduce accidental contact. A sterility control contains uninoculated medium treated in the same way. Growth in that control signals contamination. Even when the control remains clear, visible growth in the test vessel does not prove purity. Colony appearance, microscopy, biochemical behavior, or molecular identification may be required. Therefore, asepsis is not merely cleanliness. It preserves the validity of the experimental conclusion and protects laboratory safety. To design a medium, begin with metabolism. Carbon supplies the framework for cellular molecules, while an energy source drives biosynthesis and maintenance. In many heterotrophic bacteria, an organic compound can provide both carbon and energy. Nitrogen is needed for amino acids and nucleotides. Phosphorus contributes to nucleic acids, phospholipids and energy transfer molecules. Sulfur contributes to particular amino acids and cofactors. Potassium, magnesium, calcium, iron and trace elements support enzyme activity and cellular structure. Water is the reaction medium and suitable osmotic conditions protect the cell. Some organisms cannot synthesize particular vitamins, amino acids, or other growth factors and must receive them from the medium. A medium is therefore selective even before a special inhibitor is added. Its composition determines which metabolic capabilities are supported and can also alter growth rate and product formation. Two classification questions must be kept separate. First, what is the chemical composition? A defined or synthetic medium contains specified chemicals in known amounts. A complex medium contains ingredients such as extracts or digests, whose exact molecular composition is not fully defined. Second, what is the purpose? A selective medium contains conditions that favor some organisms and inhibit others. A differential medium reveals visible biochemical differences, often through a color change or colony appearance. An enrichment medium increases the proportion of a desired organism in a mixed sample by providing favorable conditions. These categories can overlap. A medium may be complex, selective, and differential at the same time. Growth on a selective plate supports compatibility with those conditions, but it does not independently establish identity. A strong answer states both the medium property and the evidence needed for confirmation. Sterilization aims to eliminate all viable microorganisms, including resistant forms, from the treated material. Moist heat under pressure, as in an autoclave, exposes materials to saturated steam at a temperature above the normal boiling point of water. The pressure enables the higher temperature. The lethal action comes mainly from heat and efficient steam contact, which damages proteins and cellular structures. Dry heat transfers energy less efficiently and usually requires higher temperature or longer exposure, but it is useful for suitable glassware, metal, or materials incompatible with moisture. Success depends on the actual time at the required temperature throughout the load, not only the instrument setting. Air pockets, overpacking, or poor steam penetration can cause failure. Chemical indicators show exposure, while suitable biological indicators provide stronger evidence that the sterilization process achieved its intended performance. Not every material can be heated. A heat-sensitive solution such as a serum component may be passed through a membrane that physically retains microorganisms. The filtrate can then be collected aseptically. High-efficiency filters remove particles and microorganisms from air in suitable systems. Radiation may damage microbial nucleic acids and can treat selected surfaces, rooms, or disposable materials depending on the radiation type and dose. Chemical agents are chosen according to target organism, concentration, contact time, interfering organic matter, and surface compatibility. The vocabulary matters. Sterilization has the most complete objective. Disinfection reduces or eliminates many harmful microorganisms on inanimate objects, but may not destroy every resistant form. Antisepsis applies suitable chemical agents to living tissue. The correct method is selected by asking what material is being treated, what level of control is required, and how success will be verified. A pure culture contains one kind of microorganism, so cells from a mixed population must first be separated. In street plating, an inoculating tool carries cells across successive regions of agar. Repeated dilution over the surface eventually deposits separated viable units that form isolated colonies. In a spread plate, a measured diluted sample is distributed over the agar surface. In a pour plate, the diluted sample is mixed with molten, suitably cooled agar, so colonies may develop within and on the medium. Serial dilution may be used before either quantitative plating method. A colony is described as arising from a colony-forming unit, because the starting unit may be a single cell or a clump. Picking one well-isolated colony improves the chance of purity, but confirmation can require repeated streaking, consistent colony morphology, microscopy, and biochemical or molecular tests. Separation is the beginning of verification, not its end. Each microorganism grows within a range of physical conditions, with a minimum, optimum, and maximum that must be stated for the particular organism and experiment. Temperature changes reaction rates, protein stability, and membrane properties. Outside the tolerated range, metabolism slows or cellular damage increases. pH affects ionization, transport, and enzyme function, and cells may regulate their internal conditions only within limits. Oxygen is essential for obligate aerobes, harmful to obligate anaerobes, and used flexibly by facultative organisms. Tolerance depends partly on pathways that handle reactive oxygen species. Water activity describes available water rather than total water. High solute concentration can draw water from cells and inhibit growth. These factors interact, so an optimum measured in one medium may shift in another. Experimental comparisons must change one stated variable, hold other conditions constant, and measure growth using a suitable method. Plot the logarithm of cell number against time for a closed batch culture. During log phase, cells adapt to the new medium, repair damage, and synthesize components, metabolic activity may be high, even though population increase is limited. During exponential or log phase, cells divide at an approximately constant maximum rate under the stated conditions, so population size increases geometrically. As nutrients become limiting and wastes accumulate, net population increase slows, and the culture enters stationary phase. Cell division may be balanced by cell death, and stress responses or secondary metabolite production may become important. During decline or death phase, the number of viable cells falls, though the pattern need not be perfectly exponential. The phases describe population behavior under batch conditions. They also explain why cells collected at different times can differ in physiology, gene expression, product yield, and sensitivity to treatment. During ideal exponential growth, a population that divides by binary fission doubles in each generation. The relationship is n equals n0 multiplied by 2 to the power n, where n0 is the starting cell number, and n is the number of generations. Taking logarithms allows n to be calculated from starting and final populations. If total elapsed time is t, generation time g equals t divided by n. For example, if a population increases from 1 million to 8 million cells, the increase is 3 doublings, because 8 equals 2 cubed. Over 90 minutes, the generation time is 30 minutes. This calculation assumes exponential growth and equivalent division behavior over the interval. It should not be applied blindly across lag, stationary or decline phases. Temperature, medium composition, aeration and strain all influence the observed rate, so conditions belong with every reported generation time. A direct microscopic count estimates particles or cells in a known volume, but unless viability is assessed separately, it may include living and dead cells. Turbidity or optical density measures how strongly a suspension scatters light. It is rapid and useful for following growth, but it is an indirect estimate, becomes non-linear at high density, and requires calibration for the organism and instrument. Dry weight measures biomass and is useful when cell counting is difficult, but it also does not directly measure viability. A viable plate count estimates colony-forming units from cells able to reproduce under the chosen incubation and medium conditions. It can underestimate cells that are injured, clumped, or unable to grow on that medium. Therefore, these measurements may diverge during stress or decline. A complete interpretation names the quantity actually measured, rather than simply reporting that the culture has more or less growth. Serial dilution converts a dense sample into plates with separable colonies. Suppose 1 milliliter is transferred into 9 milliliters of diluent repeatedly. Each step is a 10-fold dilution. If 0.1 milliliter of the 10 to the minus 5 dilution produces 60 colonies, the estimated original concentration is 60 divided by 10 to the minus 5 multiplied by 0.1 milliliter. This equals 6 times 10 to the 7 colony-forming units per milliliter. Write the dilution and plated volume explicitly, so the powers of 10 are not guessed. Select a plate within the method's accepted countable range. Use replicate plates where possible and report uncertainty. The answer is CFU, not necessarily individual cells, because a colony can originate from a clump. Sampling error, mixing, pipetting, plating efficiency and incubation conditions all affect the estimate. Culture maintenance aims to retain a living, correctly identified, and stable biological resource. Frequent subculture can keep organisms available for short-term work, but repeated passage consumes time and may select variants or introduce contamination. Refrigeration slows metabolism for suitable organisms and periods. For longer storage, cultures may be frozen with an appropriate protective agent or freeze-dried when the organism tolerates the process. The best method depends on organism, purpose, and validated recovery conditions. Every stock needs an unambiguous identifier, organism or strain information, date, passage history, storage location, and responsible person. A working stock can be derived from a protected master stock, so the original is disturbed less often. After recovery, viability, purity, and important characteristics should be checked. Successful preservation is not merely survival, it includes authentication and retention of the properties for which the culture is maintained. Biosafety begins with risk assessment. Consider the organism, concentration, culture volume, route of exposure, aerosol generating steps, and consequences of accidental release. Appropriate containment combines laboratory design, good microbiological practice, protective equipment, and equipment such as a biological safety cabinet when required. A cabinet is not a substitute for correct technique, and an open flame is not appropriate in every controlled airflow environment. Work surfaces and waste require validated decontamination, spills require a defined response, and cultures must be transported, stored and disposed of through authorized procedures. Clear labels and records protect traceability. Quality control also supports safety. Contaminated or misidentified cultures can invalidate research and create unexpected hazards. Always follow the institution's approved protocol and trained supervision. A school-level explanation should describe principles and never improvise procedures for unknown or potentially harmful microorganisms. For a 3 or 5 mark CBSE answer, begin with the exact objective. If asked about media, separate composition categories from functional categories and give a reason for each. For sterilization, distinguish the purpose of pressure from the lethal action of heat, then mention time, penetration and indicators. For pure culture, write separation, isolated colony selection, and conformation. Draw the growth curve with labeled axis and lag, exponential, stationary and decline phases. Explain the physiological reason for each transition. In the viable count calculation, show dilution, plated volume, colonies, units, and the CFU limitation. When comparing measurement methods, state whether the result reflects total cells, viable CFU, turbidity, or biomass. End experimental answers with a sterility control, purity check, replication or biosafety boundary. Never claim organism identity from color or visible growth alone. Close your notes and rebuild the chapter. Microbial culture provides defined conditions, while asepsis prevents contamination and exposure. Nutrient requirements reflect metabolism, and media are classified separately by composition and purpose. Moist heat, dry heat, filtration, radiation and chemicals are selected according to material and control objective. Streaking, spreading, pore plating, and dilution separate viable units, but an isolated colony still needs conformation. Temperature, pH, oxygen and water activity define growth ranges. A batch culture passes through lag, exponential, stationary and decline phases. Direct count, turbidity, biomass and viable count measure different quantities. Serial dilution and plated volume are both required to calculate CFU per milliliter. Preservation must protect viability, identity and stability, while biosafety requires risk assessment, containment, decontamination, documentation and authorized disposal. If you can connect every observation to the correct inference and limitation, you understand microbial culture.